வணக்கம். Welcome to this course on classics and neuroscience. In this series of videos, we are looking at classic Nobel Prize winning works. In this series of videos, we will look at classic Nobel Prize winning works of Hodgkin and Huxley. For want of time, we will only look at one of the 5 impactful papers, 5 legendary milestone papers by Hodgkin and Huxley. We will only look at one of them. In future years, I propose to describe in more detail the other papers and review the work that was done using other papers. Here, I will describe what the 5 papers were about, the landmark 5 papers, what are they about, I will briefly describe, and I will discuss one paper in brief detail because of time constraints. So, in this video, we will look at a series of 5 landmark papers by Hodgkin and Huxley, and we will only look at the first paper. These are the 5 papers proposed to be discussed in this course, but for now we will only discuss the first paper. The first paper essentially described the experimental method, the behavior of the neuronal cell membrane in a normal ionic environment. What happens when the ionic environment was maintained as the physiological level? What happens and how to perform an experiment in this situation, and what is the nature of membrane potential in this case? How to do the study? What was the experimental approach? That was the description of the first paper. And in the second paper, they studied effect of changes in sodium concentration and the resolution of the ionic current into sodium and potassium currents. So, essentially this was the first brilliant insight that the changes in the membrane potential perhaps happens due to changes in the ionic currents, the permeability in the units of conductance to specific ions, conductance to sodium ions, conductance to potassium ions, was a brilliant insight. The first stroke of brilliance that this membrane potential or this changes in the membrane potential was perhaps due to this dance or balance or the lack of balance between sodium current and the potassium current, and the way these two currents change, that essentially leads to the changes in the membrane potential on the neuronal cell membrane, was a brilliant insight. So, that is due to the changes in the permeability of the membrane due to specific reasons and at specific times. Also, there is a need to study this across the membrane because you are interested in studying this communication that happens along the neuronal cell membrane. So, there is a need to study the changes along the cell membrane. So, there is both a spatial and temporal distribution. So, there is a change that happens due to time, and there is a change in the ionic channels that happens as a function of the length of the or the location of the neuronal cell membrane. So, and suddenly if you change the membrane potential, what happens to the ionic conductances? That is the third paper. And another brilliant insight is the description of an inactivation process that suddenly reduces sodium conductance or sodium permeability during the falling phase. So, the action potential has a rising phase and a falling phase. This is the rising phase and this is the falling phase. During the falling phase, the sodium conductance drastically reduces. Actually, the falling phase of the action potential is because of the reduction of the sodium conductance, but that is something that I now know after these studies. But that causal interpretation came essentially from Hodgkin and Huxley. And essentially in the fifth paper they concluded the series and showed that the form and velocity of action potential may be computed from the results that they described previously, and they provided a set of equations that could describe this neuronal communication. So, perhaps the most important contribution, one of the most important contributions is to describe the nature of membrane potential or the change in membrane potential as a function of change in the conductance, ionic conductance. They chose the squid giant axon for performing this study because these axons were very large in diameter, about half a millimeter to 1 millimeter broad, that made it absolutely ideal for performing experiments. Quite large, 1 mm in neuronal terms is actually very large, and this provided an opportunity to insert an electrode inside the neuronal cell membrane without damaging the tissue. So, you can insert an electrode inside and keep a reference electrode outside to measure the potential difference, very important. And also you can have a stimulating electrode, another electrode that sends current inside and changes the potential. So, you can clamp the voltage, clipping, clamping, clipping, clamping. What do these things mean? In basic physics and electrical engineering, please do check using Google or ChatGPT. Learn through your own efforts what does voltage clamping even mean. Okay, essentially you are raising the potential level of the membrane briefly. So, this squid giant axon essentially provided a robust model for studying action potential dynamics, and a brilliant choice of model system. So, several things worked well for Hodgkin and Huxley. One is the presence of the method, the other is an excellent experimental model system. This is absolutely crucial. You can have a brilliant approach measurement system, but if your selected level of analysis and if your selected organism or if your selected model system is not helping you achieve your goals, then you are going to struggle a lot. So, that is another thing that worked exceedingly well. Perhaps they also worked with several species before and landed up with this one after a lot of trial and error. The details are left for you to check yourself. So, in the first paper, they essentially described the current voltage relations in the membrane of the giant axon of Loligo. This picture is the Loligos, this is not the neuron, this is the entire Loligo. So, in this paper, they describe the experimental method, the behavior of the neuronal cell membrane in the normal ionic environment. So, they had two fine silver wire internal electrodes, and they had a gauge system to measure membrane current, and they had a feedback amplifier that will allow them to voltage clamping. This voltage clamping was a stroke of brilliance from these two brilliant scientists, so that they can change the voltage level of the membrane and then see what is the reaction of the membrane. So, one is measurement electrode, the other is the stimulation electrode. So, you can stimulate, you can send a current inside and then see what happens as the reaction. This is what Sherrington did, but not for individual cells. Recall, this is perhaps what Sherrington was doing, this is what was performed by other classical and brilliant neurophysiologists of the time. For example, similar things, Keith Lucas, Edgar Adrian and other brilliant neurophysiologists, electrophysiologists of the time were doing this, but they were not doing this at the individual cell level. Hodgkin and Huxley were doing this at the individual cell level, and what they found was that there was an all or nothing phenomenon that happened within the cell. So, 100 millivolts of change in the potential, membrane potential, when you stimulate with a brief shock, a sudden change like that, a spike. with a threshold of approximately 15 millivolts. Note that this will change between neurons, between types of neurons, between organisms. They are describing this in the Loligo in the model system that they were studying. And there was graded response. So, if you provide a small stimulus below 15 millivolts, you will get a small response. If you provide a slightly larger stimulus than the previous one, but still below 15 millivolts, you will get a slightly larger response, and so on and so forth. Do check my other course on Neuroscience of Human Movements, in which I provide a detailed description of what is a graded potential, what is an action potential, what are the differences. I describe this for nearly half an hour or 45 minutes. The details are left for you to read by your own. And they found that depolarization at below 15 millivolts caused outward currents, and if you depolarize to a level above 15 millivolts, then there was an initial inward current. Inward current is that current in which cations flow inside the cell from outside. Okay. These details are also present in my other course on Neuroscience of Human Movements. So, this causes an initial inward current. After a brief period of time, there is an outward current. So, what did they do? So, they measure the membrane potential and compare it with the desired value, and if there is an error, then they use the stimulating electrode. So, this is the current passing electrode or the stimulating electrode. This is the recording electrode. This is the recording electrode. So, this is the recording. Oops. That is the recording electrode, and the one in green here is the one in black here is the stimulating electrode. Okay. The one in red is the recording electrode. Let me mark the stimulating electrode in yellow here. Okay. That is the stimulating electrode for the sake of this. And they checked whether there is a, this is the control circuit that checked whether there is an error in the voltage. If so, a current is passed through the stimulating electrode. Does it cause a change in the ion channels? If yes, then they again check whether there is an error in the potential, and if it does not cause, then again they measure the membrane potential. So, this essentially gave them an opportunity to artificially change the membrane potential and measure the responses at various input levels of membrane potential. At various stimulating levels, what is the output of the neuronal system? So, this is a classic approach. So, you give an input and you study the output. So, are there critical input voltages at which some critical events happen? So, they found that there is an inward current that happens when the input voltage was more than 15 millivolts. That is, when you clamp with more than 15 millivolts, suddenly there is a huge amount of inward current that happens, and they later described this due to a change in the sodium conductance. That is what they found that. So, after the depolarization went to 110 millivolts, this inward current disappeared and was replaced by an outward current. So, they essentially found the relationship between the ionic current and the membrane potential. Most importantly, they were also able to study this using a slightly different reagent, and the effect, how does this effect change in the slightly different reagent. So, essentially they were able to attribute these changes to the presence of specific ions in the extracellular matrix. So, that is critical, that this is happening, that the inward current is happening due to the presence of excess sodium in the extracellular matrix was the brilliant insight that they could make. So, this relationship essentially happens or described both in the rising phase of the action potential. So, there is a relationship between the ionic current and the membrane potential, and the maximum inward and outward ionic current was temperature sensitive. So, if you change the temperature, if you increase the temperature, the rate of change, so that means that these are also sensitive to temperature. And when you compensate this error with feedback, this was due to the presence of impedance or sort of a resistance induced due to the membrane capacitance. The details of this I am skipping for want of time. Here is the input that they saw. So, this is it, and when you stimulate by 15 millivolts, there is an action potential of this type, and this is due to the inward current. After some time, this inward current disappears and you only have outward currents, and during this time they found better sodium conductance, and during this time they had low sodium conductance, critical. Okay. They further followed this up with their other papers. With this we come to the end of this video. With this we also come to the end of this week's content. Hopefully in the next offerings of the course, we will be able to describe the other papers by Hodgkin and Huxley for this week's content. Thank you very much for your attention.